Okay, welcome. Welcome to our last session. It's our 7th session, which goes very quickly. And today we're going to be focusing on semiochemicals for fall armyworm monitoring and control. My name is Alison Watson, and I'll be moderating the session. And before we start, I just want to say a big thank you to all our key support organisations, obviously our co-hosts, Cabby. Thank you for being there with us throughout this journey. And also our partners, DFAT, the ASEAN Secretariat, Ministry of Agriculture and Rural Development, Vietnam, and CSIRO, which is the host organisation of the ASEAN Fall Armyworm Action Plan. I'm also just going to say a big hello from Pheung. He's the lead for the Cabby Southeast Asia Group team. He's actually recovering from COVID and he couldn't be with us today, but that's a shame. But he did actually pass on his thoughts, and welcome to everyone as well. Now, just a reminder, you all know this, the session will be recorded. We will post a copy of the presentation afterwards on our website, and we'll also send you a email just as a reminder of where to find that. Today, if you'd like to interact, and we really encourage you to do so, please use the Q&A box for asking your questions. It really helps us to moderate the session efficiently. And if we don't have time for all your questions verbally, it allows the presenter to also write in some answers by writing while the other speaker is speaking. So it is quite helpful. Please also use the chat box to introduce yourself. We're pretty good at this, but please say who you are, maybe what organisation you're working for, which country you're in, and share that with everyone because it helps build our collaborative network. And also please share any information, any research you're doing, or interesting work that you are working on at the moment. Now, as I said, this is our last session for the year. Incredibly, it has gone very fast. 13th of December, so the calendar year is almost up. I know everyone is very busy, so thank you for joining us, all 88 participants, so that's a fantastic number at this time of the year. Over the next week, we're going to be putting together a highlights package of all the different webinars that we have run, and we'll also be releasing a training video on the use of BT biopesticides, so we're very excited about that and we'll be in touch. Now, today we have two really fantastic speakers. We've decided to just really have two really good speakers who are going to really spend the time stepping you through their research and their work, giving you a bit of background to what's happening in Vietnam as well on fall armyworm, but really exciting research from Vietnam that Dr. Phoung will be sharing with us. And then Rob is going to take us through trapping for fall armyworm. It does sound a bit of an adventure out there at night, as he's explaining to me when he goes out trying to find these fall armyworm. So I'm excited to hear about that as well, because we haven't really talked about that before in any of the previous sessions. And I think it'll be a very practical, both presentations, a very practical session for you. So please ask lots of questions of both experts. Now, what I'm going to do now is I'm going to run a poll. It's just a quick poll. It's not very difficult. And it really is just to sort of warm us up, see what we're thinking. First, just to have an idea of who is in the room. Have you worked with or researched semiochemicals for fall armyworm control or monitoring? I'm thinking of the big picture there. And we're already racing to a start there with lots of people answering. And then an important tool for monitoring fall armyworm is pheromone trapping. Does the design of the trap or lure make a difference to how many fall armyworm are caught? Okay, so we'll just have a bit of an idea of what you say. Could be a trick question, could not be, just to bring in a little bit of ambiguity into that question, make it a bit harder for you. We've got 47 people of 94 people in the room now. So I'm going to see if I can get up to over 75. 80% is my gold standard, but we're getting up there actually. I know that if you're logging in via a phone, it can be really hard to actually participate in the questionnaire. So sorry about that, but you could tell us your answer in the chat if you'd like to. I need a few more people because I've only got 67% participated. Maybe a few more, we can push it up a little bit. And this will give a good idea to both our speakers today, just who's in the room and how many questions they're going to be asked, maybe. So I'm almost at 70%. Couple more people. If you haven't answered yet. Oh, here we go. So I'm going to end the poll. I'm going to share the results. And so, have you worked with or researched semiochemicals for fall armyworm control and monitoring? We've got 32% yes, 60% no, and 7% no, but I will in the next 6 months or so. So there we go. You've got a bit of spread of talents there and experience and expertise, Rob and Pheung. So you'll be able to sort of know that you may get some really difficult questions and you may get some questions around how do I get started as well, which I'm sure both of you will cover. Now, an important tool for monitoring, does the design of the trap or lure make a difference to how many fall armyworm are caught? This is going to be really pertinent for Rob. We've got yes, 57%, no, 6%, maybe 18%, I don't really know, 19%. So there we go. It's going to be very interesting, and you'll probably find out the answer to that in Rob's presentation later. Now, let me just get rid of that. And I'm going to move on to the first presentation. Now, Pheung had a little bit of problems, Dr. Pheung had a little bit of problems with her sound, so we're hoping it's all good. We had a bit of a panic, but look, it's wonderful to have you join us, and you're doing some very exciting work and very successful work in Vietnam. And so it's a real privilege to have you join us today. And I've had the real privilege as well of visiting Dr. Pheung in Vietnam, and it was truly a pleasure and very exciting for me. So I'm very pleased that she could be here with us today. So thank you very much. And you just tell me when you need to move to the next slide.
Okay. Thank you, Alison. Good afternoon, everyone. Today I'm very happy to share some of my research and applied pheromone trap in IPM to control Pheromones in Vietnam. Next slide, please. And currently, I research about the semiochemical, including the pheromone, and alarmone, for the control the plant management. The most important research I conducted that is a female sex pheromone. And next slide, please. Research about the female sex pheromone in the Lepidopteran species, there are the main two types of female sex pheromone were separate, and another type of female sex pheromone would not belong to type 1 and type 2. And you can see the Lepidopteran can release many kinds of pheromones, and we can apply the female sex pheromone to control and monitoring the main moth on the field. And next slide, please. Research about the female sex pheromone of some insect pests on maize. In Vietnam, we have several Lepidopteran species that is on maize, and they have a female sex pheromone, and the components have a difference between species, and we can apply the female sex pheromone to control each species. And currently, I research about the female sex pheromone of Asian cornborer, ear cutting, black cutworm, straw armyworm. And the most successful currently I obtained that is a female sex pheromone of tobacco cutworm and straw armyworm, and we already test on the field. Next slide, please. For the straw armyworm in Vietnam, from 2019, straw armyworm present endemic on maize in Vietnam around 76,000 hectares. And in 2020, the area of maize was infected by straw armyworm was significantly decreased to 29,000 hectares. And 2021, the area of maize was infected by straw armyworm also significantly decreased, only 15,000 hectares. And in 2019, The year loss by Pheromones was estimated about 50% if we don't apply the control any methods to control on the field. However, currently our our farmer can control the pheromone very good, and the loss will significantly decrease. Next slide, please. After pheromone present in Vietnam, we also record the host plant of pheromone in Vietnam. Although this species was recorded that they present on over 353 host plant species. However, in Vietnam, we already record this species on maize, paddy rice, onions, and elephant grass. However, the main host this species that is maize. On the paddy rice, not too much pheromone damage on paddy rice. And the second that is natural animal pheromone in Vietnam, we also record some parasitoid wasp, insect predator, and some fungi that is a national enemy of pheromone. Next slide, please. Here is some photo of parasitoid wasp pheromone we collect on the field. That is Telenomus remus and Telenus trichogramma and microbletis marinae. That is some parasitoid wasp we record in Vietnam. Next slide, please. And the next is some predator and insect pathogen pheromone were recorded in Vietnam. And on the field, this species can control the larvae of pheromone very good on the field. Next slide, please. And another research we conducted that is a research about the morphological characteristics of pheromone. And the morphological characteristic of pheromone is the same with all the morphological characteristics of pheromone in another country. No significant difference. And another, we compare the damage symptom of pheromone in Vietnam from early leaf, word leaf, young flower, and year corn. So you can see in the photo that's the damage of pheromone on the maize in Vietnam. Next slide, please. About the host strain of pheromone, we researched to identify that they have a two host strain, that's corn strain and rice strain. There is a difference between two host strain of pheromone in Vietnam based on local site, restriction enzyme, and female sex pheromone. And the restriction enzyme, we can easily compare the difference between the corn strain and rice strain. And also we use the ratio of pheromone components between the four main components to identify the two host strain of pheromone in Vietnam. Next slide, please. To compare, to control the pheromone in Vietnam, we need to identify the development time of pheromone in Vietnam in the different temperature. And we also we got the result that the life cycle of pheromone very short, around 20 days in the summer. So the number of larvae on the field very high and the life cycle very short. So in the summer, very difficult to control this species. Next slide, please. Because in the summer, the density of the pheromone on the maize very high. So the control and management of pheromone a little bit difficult compared with the spring and autumn. And in Vietnam, we have conduct some training course for the farmer and for the staff of the local staff to how to control the fall armyworm. And after the training, they will return to the local and they will training for farmer again. And there are some training program on the IPM for the local staff. Next slide, please. For the management of fall armyworm in Vietnam, we apply the integrated pest management and we apply the 5 methods. First is the cultural control. Second is the host plant resistance. Third is the pheromone control. The fourth is the biological control. And finally, that is the chemical control. And next slide, please. And there are some photos of the IPM IPM practice and IPM model in Vietnam. Farmer was joined with the model for the IPM, and you can see from the start of May season until the end of season, and they can practice on their May field from the beginning of season and the end of season. And next slide, please. For the first is a cultural control. In Vietnam, we apply several methods. For example, intercropping bean and maize, intercropping peanuts and maize, and the third is a crop rotation of paddy rice and maize. These methods can significantly decrease the density of larvae on the maize. And almost this model, the farmers don't need to apply the chemical control for fall armyworm. Next slide, please. And the second is a host plant resistance. In Vietnam, the farmer can apply some by various resistance to fall armyworm. And if the farmer apply the by variety resistance to armyworm, they also don't need to apply the chemical control. Next slide, please. And the most important my presentation today, that is the pheromone control. The pheromone control is the key of the IPM model in Vietnam. The pheromone control we apply for the 3 subjects. That is, the first is monitoring of fall armyworm on the field. The second is the map trapping moth of fall armyworm. And the third is the mating interruption of the moth of fall armyworm on the field. And both mating disruption and mating confusion we also test on the field. And both of these methods have a good result to control the fall armyworm on the field. Next slide, please. So first, for the apply the pheromone control, we identify the female sex pheromone of fall armyworm in Vietnam, and we compare with which is the pheromone component is the high heat or the main component or strong is bioactive, and we prepare the pheromone loop. And in our research, we use the 4 female sex pheromone of 4 female sex pheromone component of fall armyworm to prepare the pheromone loop. Next slide, please. Next slide, please, Anderson. And next slide, it is the result. We test pheromone loop on the field. We compare some pheromone loop type, including 2 components, 3 components, and 4 components. And you can see the result. That is 4 components, 3 components, and 2 C2. That is have a number 2 have a high heat of male moth capture on the chaff per night in compare another treatment. And after this test the pheromone loop on the field, we will apply 3 treatments for the next research on the field, and we also apply 3 treatments for the IPM model. Next slide, please. For the pheromone control, our team also conduct some research about the pheromone trap type. We will compare the ability to capture pheromone, male moth in the trap, and the color of the trap have a high heat. The male moth on the trap that is yellow. In a compare with the commercial pheromone trap, beside the commercial bucket tunnel trap, we can use our handmade trap with a yellow color. The number of male moth in the trap significantly higher than another color, and the farmer can apply. The handmade trap very cheap compared with the commercial pheromone trap, and this trap also was applied in several provinces and have a good result. Next slide, please. About the method applied the pheromone control in Vietnam, we applied the distance between the trap from 20 meters to 30 meters, and the height of the trap around 1.2 meters to 1.5 meters. So the height of the trap will change when the height of the maize change. When the beginning of the maize season, we place with the height around 1.5 meters, and after that we will move to 1.5 meters. And the time apply the pheromone trap on the field from the beginning of the main season at the grow date until the 2 months later, around the young year corn appear. That around 2 months. That's the time to change the pheromone loop is 30 days. So in the one maize season, we apply 2 times. We change the pheromone, or we apply 2 times. So the total time apply the apply the pheromone trap on the maize season. One maize season is around 6 days, around 2 months. Next slide, please. And you can see the result of the total main moth capture on the pheromone trap in the figure A. In the figure A, the male moth capture on the pheromone trap significantly higher in the first week. After 4 days, the number of male moths capture on the trap total around 1,200 male moths. And after that, the number of male capture in the trap significantly decreased. And around 10 days later, the number of male moths in the trap also very significant, lower than the first 1 week. And the figure B, that's the density of Pheromone worm on the maize plant. So in this figure, we compare between the normal maize and the maize resistant to the insect. And in the blue lines, that is the normal maize, the density of Pheromone worm in the second week very high. However, after that, the density of Pheromone worm significantly decreased, and when the density of larva of Pheromone worm around 16 larva per 1 square meter, the farmer will apply the chemical control, and after that, the density will significantly decrease, and the density equal with maize resistant to insect. And the second, after that, the density of pheromone a little bit increase. And the second time, the farmer will apply chemical control around in the 18th of October. And in this May season, the farmer only applied two-time two-time chemical control. So after that, the density of the pheromone significant decrease and not different compared with the recent Mays. And next slide, please. And another method we research and apply in the IPM model that is biological control. In this method, we apply some insect pathogenic fungus, bacteria, and virus that already have a commercial product, and the farmer can buy it and apply on the field. However, this method needs apply when the larva is very young, first or second instar larva. If the larva 4th or 5th instar larva, this method is not highly effective to the larva. Next slide, please. Besides that, in Vietnam, we also apply some extract, for example, ginger, garlic, and chili. Ginger and garlic, and neem oil, ginger, garlic, and chili to apply to control the pheromone in Vietnam. However, this extract needs to apply when the larva pheromone very early stage, first or second instar larva. Next slide, please. And finally, when the density of pheromone too high, and we need to apply the chemical control, we will apply some chemical that plant protection department encourage the farmer apply. And these chemical control can help the farmer to control pheromone very good in Vietnam. And next slide, please. And finally, that is in Vietnam, many provinces will apply IPM for pheromone in Vietnam, and the farmer will have applied the thermal control from the beginning of the May season, and they will follow all steps of IPM by the plant protection department encouraged, and we were success to control in the Vietnam. And the most important that we reduce insecticide apply on the field from the 4 to 5 times to 1 or 2 times per 1 May season. And the economic efficiency in the IPM model has increased from 17 to 25% compared with outside of the model. Next slide, please. Thank you very much for your attention. That's my some our research and some results on the IPM in Vietnam, and also research about the pheromone and apply the pheromone in the IPM in Vietnam. And thank you so much.
Thank you very much, Phoung. Look, that was fantastic. I apologize to those that may not have heard so well. I could hear you, but there was a bit of a strange sound in the background sometimes, but I could hear everything. And you have quite a few questions here as well. So Phoung, I'm really interested. You have had a lot of success with your pheromone traps with farmers. Can you explain it? Are farmers excited, or are they interested in using your pheromone traps?
Thank you very much for listening. When we conduct the research on pheromone on the field, the farmer very interested to join with us, and they want to apply the pheromone trap on their field. And for example, in the one province, in the one 1 year they have two main seasons. In the spring season, we apply the pheromone trap for on the May field only 5 hectares, and the second season is the summer season. We come back again, and the farmer asks us that help them to place the pheromone trap again, and they want to place pheromone trap whole of the May area of them around 20 hectares. So because in the spring season we apply the pheromone trap and they don't need to apply the chemical control. In compare with another area, they didn't apply the pheromone trap. They have to apply 4 times chemical control. So so they see that if they have a pheromone trap, they don't need to apply the chemical control, and they require that please help me to place pheromone trap on their field, and please make more pheromone trap for them. And they're very happy when we come back again and place the pheromone trap on the field for them. And in the spring season, we apply only 5 hectares, and the summer season we come back and place all of the pheromone trap for 20 hectares.
20 hectares, yeah.
Yeah.
That is great. It's great when farmers can see something in action, and also they're enthusiastic about it. And I think last time you told me that some farmers were purposely going in and shifting them to their to their little plot of land.
Because when we come back and place the pheromone trap on the field, and the farmer we decide to place the pheromone trap on the area, but the next area they don't have a pheromone trap. They ask us please move pheromone trap to their field. One is okay.
Phoung, really great results when you sort of combine all those tactics for IPM with your pheromone traps, etc., and disruption technology. And I guess what was really startling was that ability to sort of go down from 4 to 5 applications of pesticides to sort of 1 to 2 applications per season. That's a really really positive outcome. So well done to everyone in Vietnam that's working on that. I've got a question here around, can people get your pheromone traps? Can they access your technology, your solution? And I understand you've already maybe sent some to the Philippines to test there. Is that right?
Yeah. And last week I already sent some pheromone to the Dr. Barbara from in the Philippines to test my pheromone. And they in a previous workshop, Dr. Barbara told me that they bought pheromone from the USA, and they don't have a pheromone. So I thought that I can share my result, and I can share my pheromone to the Philippines, and they can test on their country, in their country, and share the result with us. And last week I already sent to Dr. Barbara, and three type of pheromone for 3 species. So I hope that in the near future I will got the result from Philippines about the pheromone test on the field.
Excellent. Thank you. Dr. Phoung, what we'll do is we're going to move on to have Rob's presentation, but we may come back to you and we might have time for discussion at the end. You actually have quite a few questions in writing in the Q&A box. I'm not sure if you can access that, but if not, we will come back to them. And I see Rob has been kindly helping to answer some of those as well. So he's very industrious at the background helping out. If you can access them, if you could answer a few of them, that would be very helpful, Dr. Pheromones. But thank you very much, and we'll come back to you. Okay, so don't go away.
Thank you, Alison.
Thank you so much, and we'll connect at the end, because I'm going to introduce Rob, because Rob's been patiently waiting there in the middle of his night. I think it's like 3 o'clock in the morning or something, but he's obviously extremely passionate about this area of research and action, and you're getting lots of claps, by the way, Phoung, like there's lots of hands going there on the screen to give you, and hearts as well. So people are very happy with your presentation. And I may try and even record it a little bit later on in the year or next year. So we've got a really clear recording for everyone, but I could hear it fantastic. So Rob, introducing you, if you could load your presentation, that would be great, because I think this is going to be a really good segue into sort of understanding design and how that plays a part, and it would probably answer some of these other questions that are popping up. So welcome to the room. And Phoung, if you want to, you can turn off your camera now if you want. And Rob, are you still around? I can see your screen. It just this needs to be enlarged.
Okay. It's not going to, there we go.
Perfect.
How's that?
Yeah, it's looking great. Thanks, Rob, and welcome to to our biocontrol workshop series. You're lucky last presenter of the year, actually. So that is not the pole position, but it's probably one of the most important positions. You've got big boots to fill.
Well, thank you, Dr. Watson, for inviting me. Yes, it is the kind of the middle of the night, but I actually do field work in the middle of the night, checking traps at different times. So I kind of did the same thing for this. What I want to talk about today, and Phoung did a really good intro to pheromone trapping. I'm going to talk probably a little more from a Western Hemisphere point of view of things, and certainly more of a historic point of view, since we've been trapping for fall armyworms since the late 1960s. I'll talk about trap designs, pheromone composition, some of the commercial lures that are available, how habitat actually influences trap catch and influences which host strains you get. I'll talk a little bit about management. We don't really do much with using pheromones as a management tool, but there is some work that's been done. And then at the end, I'll talk about what I call extreme monitoring. And this is where we monitor not on a local basis, but on a fairly wide geographical basis. So these are some of the traps. Again, Phoung had talked about the standard unit trap or bucket trap. There's sticky traps that are available that have a insert that you can pull in and out, and they're cheaper. They're much easier to hang. You can hang them on almost anything. Historically, in the U.S., especially in the central U.S., they used these large wire cone traps, and these were very successful, but it $200 to $250 US dollars to construct, so you can't have a lot of them, and it's just a really hard trap to move. There is a fabric version of these, but I'm still not a fan. However, I've done some work with cooperators in West Africa. This is one of their, I call it a Togo trap because that's who worked on it. It's essentially a Coke bottle that's been cut, and the lure is put right there. And then in Benin, they have what they call a jar 2 trap. And again, it's a little more elaborate than this one, but you can see the funnel and the lure is right there. And I'll talk about how these do in trapping situations. So for me in Florida, I've trapped since the early 2000s, comparing different traps, the unitrap and the Heliothis cone trap. The unitrap or bucket trap always does better. Doesn't matter what the lure is. They always collect more moths than the cone traps. And as Pheung was talking about the color of the traps, the color is actually really important, surprisingly. You think a moth, okay, all they're interested in is the pheromone plume, but that's not true. And if you go out at night, you can see why. These standard traps that have the green top, yellow funnel, and white bucket are kind of reflective, and the moths do use some vision to orient to the trap. All white traps and all green traps don't do as well, and you can see over time moths per night, many more moths in the standard traps than in these traps. What I will say is the all green traps are very useful in an area where you have a lot of bees, honeybees or bumblebees, because they're not attracted to the traps as much. Okay, so what did the traps do in Africa? This is work that was done in Togo, and you can see the bucket trap caught almost 2 times more moths than the sticky trap or the local trap. I was actually very surprised. The local trap did really well, and of course it's very inexpensive, relatively easy to make, and so I was that was a good thing to see that these local traps that the farmers were actually putting together themselves were effective. This graph, and you'll see this graph again, this combines the trap and the lure, and you can see the first 3 bars here are all bucket traps with different lures. And I'll talk about the lures here in a second. In Benin, they looked at the jar 2 trap. Unfortunately, it wasn't as successful as a unit trap, and did relatively poorly. Now the pheromone components, and again, Pheung did a really nice introduction to this. The original pheromone that was introduced, that was described back in the late 1960s, was the correct one, was the 9,14 acetate, but it never did well in the field for some reason. So they then thought, oh, maybe it's the 9,12 acetate. So for years they were using this as the main lure, but they had to put 25 milligrams of the pheromone into the lure. And so that should have been a hint that it wasn't working, but you know, again, at the time, it's all they had. Finally, in the mid-1980s, Jim Tumlinson, the chemist, Everett Mitchell, the entomologist, combined on 2 really good papers that described, again, the most of the components that Pheung was talking about, especially the 3 of the 4 main ones here. What Tumlinson's work showed, which is interesting, and it was kind of ignored at the time and is now back, is that all you really need are 2 components, the 9,14 and the 7,12 acetate. Both of them work really well together as one lure, better than the blend, or at least as good as the blend. Everett's work showed that the septa loses pheromone fairly quickly, so you have to replace them at least every month, if not sooner. And he also showed that there's a limit to how much you put into the lure. If you put much more than 2 milligrams, it's not really helping you at all. And again, these were the traps that he used. In Brazil, the Brazilians have, for a long time, have said that the American lures, the lures made by American companies, were not successful in Brazil. Back in 2006, they came up with a E-isomer of the 7,12 acetate, and you can see here in their work that the combination of the 2 compounds, the 9,14 and the 7,12, plus the E in this lure here, did the best. And I saw in one of the questions, there's a company in Costa Rica called Camteca. They make a very good lure, and I'm not really sure what the components are. I'm guessing that this E7 might be in it, but I don't know that for sure. Some work that I've done with commercial lures, and this company isn't around anymore, but you can see there's a lot of variation trap to trap. Where you put the trap in the field is actually important. because of the way the wind kind of flows over the field, so that you get a certain location that may collect more moths than other locations. So you do get a lot of variation, but generally the main American companies collect about the same number of moths depending on the season and the location. Now, again, if we go back to Africa, what did the Togo people look at? They looked at a 2, a 3, and a 4 component. The 3 component, so the 914, the 11/6, I'm sorry, the 712 and the 11/16 lure did a little bit better. I mean, statistically better. I'm not sure based for monitoring if this is a real biological difference or not, but they showed the 3C did well. This graph you've seen again before, and again you see the 3C, the 3C, and the 3C. So again, those lures collected a few more moths than some of the other ones. In Benin, some work that was just done. It's interesting, they had no success with the 3-component lure, and I think it's because they used a recipe that they probably shouldn't have used. I think if they would have used a pheromone, a commercial pheromone company here, they would have gotten better results. But at least the 4-component blend did really well. Okay, so as I mentioned earlier, Tomlinson and Mitchell found that the 2 components, the 914 acetate and the 712 acetate, just those 2 together as a lure was really effective. And why this is important now is non-targets, and this is something that is not really looked at. So Shelby Flesher at Penn State, so that's why if you hear of a PSU lure, that stands for Penn State. He found that just those 2 components, and this is the Leucania, this is one of the non-target moths, were not collected in the 2-component lure. And that's because the Z1116 acetate is actually a major component for Leucania. So that's what they're attracted to. And you know, you can think, well, gosh, anybody can tell these moths apart. But if you get these moths in a bucket trap, boy, I can tell you, and especially this little guy, which is the same size, it's kind of hard to separate the target species and the non-target species. And that's really important. If you're doing pheromone work and you're maybe doing a threshold study or something like that, you want to count the target and not the non-target. And again, in Africa, in Togo, again, the 2, the 3, and the 4 component, the 3 component, as expected with the 1116 acetate, attracted a lot more Leucania moths than the 2 and the 4 seed. Okay, so some fun in South Florida. We were interested in how agricultural and pasture grass areas compared trap-wise or moth-wise with what we call natural areas. These are areas near the Everglades National Park and some swampy areas in South Florida, or more urban areas. This is condos that are near Biscayne Bay, just south of Miami. So one would think you're not going to find very many fall armyworm in these areas. And luckily I had a protector that kept people away from my traps. But that's not true. In the natural areas, at least in some of the traps, we got fairly good numbers. I mean, 20 to almost 30 moths per night in areas where there's no agriculture at all. So I think, especially in Southeast Asia, where you have your plots of maize are relatively surrounded, I mean, by more natural areas. Those natural areas may be keeping populations that eventually will, once you get a new crop of maize in there, will move in from the natural areas into the maize. This is the grassy area, and I'm not going to talk much about host strains in my talk today, but as expected, the small grass or R or rice strain is pretty much what we found in these grassy areas. Very few of the corn strain or C strain. Again, that was expected. However, we found something interesting in the agricultural areas. And for this study, the agricultural area was sweet corn production, which in South Florida is a major crop. So we had two, if you look at all the moths together, you have two populations, two peaks, one in the spring, one in the fall. This is very standard for fall armyworm in South Florida. It's where they overwinter. But by teasing apart the host strain in these, we found something really interesting. In the spring, the peak was both host strains, both corn and rice in both areas. And again, there was a lot of corn production in these areas, so I'm not very surprised that we got both. What we were surprised was in the fall, and this is another corn production area, we only found rice strain. The corn strain were hard to find, and we don't know where they were hiding. We looked for them, and still to this day, we don't know why we don't get corn strain at this time of year. Okay, this slide is a little busy, but let me walk you through. This is work that, again, my cooperator in West Africa, Dejema Kofi, has been trapping since 2017 for fall armyworm. And all this shows is that the habitat is important. If you have your traps near maize, you generally are going to get, except for this one exception, you're generally going to get higher numbers of moths. If you're in some of these more not minor crop as far as the farmers, but a minor crop to fall armyworm, you're going to get much fewer numbers. So the populations are there, they're just not built up to large numbers. This, we don't know why the rice, it's the only time he ever found high populations in rice, and we're not really sure what happened there. But anyways, for two different countries and several different dates, the maize areas had the more moths. Okay, so I talked about traps, I talked about the lures and the habitats. The final large area I want to talk about is management. Again, Pheung talked about mating disruption. This was done back in the 1970s and early 1980s in Florida by Everett Mitchell, my predecessor. The take-home story is it never really worked well. They put a lot of pheromone in the field. They did at times get 75 to 95% reduction in egg masses compared to fields that had no pheromone. And the way they did it was the pheromone was placed in hollow fibers. Those were all cut up into pieces, and then essentially almost like spraying a chemical sprayed in the maize. And so there were a lot of points within the maize that had pheromone release, but they just were never able to get control, and it was very expensive. So unless it was a sweet corn grower where your product, your crop is a good investment, most growers aren't going to use something like that. However, I will say now, in the last decade, there's been a lot of work done both in the US and in South America from companies, not from university or government workers, but from companies looking at mating disruption. So I suspect there will be some good products out soon. Another area of that pheromones can be used is in prediction. This is work that was done in South America and French Guiana in the middle 1980s, where they looked at pheromone traps and larval populations. There's a significant relationship here, but kind of weak. And what they were able to do, though, was use traps as an early warning system. So in the pasture grass systems of French Guiana, they were able to have a whole network of traps. Once the trap numbers started getting higher, then they could let the farmers know and tell them, hey, you need to get out in your pastures and either treat or at least take a look and see what the larval populations were. In the US, the only paper I've ever been able to find in maize that looks at plant infestation and moth numbers as far as a significant correlation is this one out of the state of Maryland in the US. Again, mid-1980s, there hasn't been anything done after that I know of. This, for whatever reason, this experiment worked really well. I've tried it myself and have never gotten anything that where the trap numbers and the field infestation had a positive or negative correlation. And then finally, with management, this is work out of South America, out of Brazil, where they used pheromone traps to trigger a management strategy. In this case, they used pheromone traps. If the trap had more than 3 moths in a week, then that triggered a release of the parasitoid trichogramma. And again, Pheung was talking about some of the natural enemies. Trichogramma is a good little egg parasitoid. And so they were able to get a really nice relationship between maize yield and the number of releases, not the number of wasps that were released, but the number of times that they made a release. And again, this was all based on their trigger was number of moths per night. Again, this hasn't been repeated that I know of, but that certainly warrants some future work. The last area I want to talk about, and I know I'm running late, I'm going to try to skip through this, is what we call extreme monitoring. And what we're doing is using traps to look at very large geographical areas. Everett Mitchell did this in the late 80s, early 90s, where he had traps put out, same lure, same traps at the same time from Quebec, Canada, Tifton, Georgia, Gainesville, Homestead, Florida, and all through the Caribbean and even into northern South America. And he was hoping there would be a pattern of movement from the Caribbean to Florida and from Florida to the Caribbean. He could never find any kind of relationship. Either the wind was wrong when the populations were high, or the populations were low when the wind was in the correct direction. So it was a nice effort, but really didn't get much information out of it. However, we've been able to do a lot more because we have genetics in our pocket. Rod Negoshi is a geneticist here. He was able, within the corn strain, to find haplotype differences on one of the genes. And so we were able to tell moths that overwinter in South Texas from ones that overwinter in South Florida. And so by having a network of traps, kind of like Everett did in the Caribbean, but we did it in the continental US, we were able to see where these moths are moving in the spring. And what we found is that the Appalachians, which are not really that high mountains, but still seem to be some kind of border, that the Texas moths move up into the central US and then move into the northeastern US. And then we have a paper coming out, hopefully in another couple of months, that shows them coming into Canada, up into Quebec, whereas the Florida moths only kind of hug the coast and for whatever reason peter out here in New Jersey and New York, and they're probably blown out to sea is what we're thinking. But we've been able to monitor, able to model this movement. John Westbrook did this in a couple of really nice papers where he modeled in the spring populations coming out of Florida and Texas. And so with this work, we were able to show that his models were correct. And my last slide, I have a lot of literature. Whoops, I'm sorry. A lot of literature in my talk. And so those were some of the citations. If you have trouble getting those citations, go ahead and email me and I'll be happy to send you a PDF. Okay, that's it.
That was fantastic, Rob. Another fantastic presentation following on from Dr. Pheromones. So very, very informative. And I could have kind of listened to that for quite a long time, actually, both presentations. So really, really great way to finish the year. Lots of questions coming through. Maybe there's just so much information in there. I was actually going to ask that question around the relationship between the number that you trap versus the population numbers in the field, but you were sort of getting to that in those last slides, and that was fascinating, as well as what you just showed, that model where those Florida fall armyworms seem to stick down there. It's obviously it's quite significant that they stay there, and the others are really going in a very large distance. So really, really interesting. Looking forward to that paper that's coming out soon. Maybe a question following on from Dr. Pheung's presentation. Have you tried yellow traps? Because she seemed to have a bit of success with that. And I saw you had white traps and green traps.
Boy, have I tried yellow traps. I raided one of the hardware stores for all kinds of spray paint of sun yellow and flower yellow. I didn't know there were that many yellow colors, and spray painted traps, let them dry for a long time so the odor didn't affect anything. And some of the traps did okay, but it's funny, the standard trap, for whatever reason, I think it's the contrast of the top with the funnel color and then the bucket. I think that contrast is important. And again, in the field, you can see those traps if it's a new moon or something, it's hard to see, but any other time of the year, you can kind of see those traps. And so I think the moths are using that as, oh, hey, the odor is coming from there. Let me head that way.
Yeah, maybe
so,
they're
yes.
learning. Maybe they're learning between traps as well, with the colors. Listen, that is actually fascinating. So the standard, tell me again, what are the standard colors normally? Is it white and?
So it's a green top.
Yeah. And
then the funnel itself that they go into is a yellow funnel, and then the bucket itself is white.
Oh, that's interesting.
And it's interesting. Again, I've been out at night way too many times, but the moths, when they're attracted to a trap, they get very close to the lure, they get very excited, and they literally bounce off that green lid and fall down the funnel. Yep. And so it's not like they flop in, they go, and you can hear them hitting off the lid. It's kind of interesting.
And is that why they get quite damaged? I mean, because sometimes when you look in those traps, as you were saying, it's quite hard to tell between the different moths sometimes. So we've just had a case of that. I won't say which country it is, but it doesn't have fall armyworm, but thought they had fall armyworm with a very specific trap, pheromone trap, but it was a close relative, not the fall armyworm. So how do you sort of prevent that from happening? I mean, you sort of talked about that.
Well, we use vapor tape, which is a dichlorophos. It's a fumigant. And so that's what we put in our bucket traps to kill the moths.
Okay.
But if you get too many moths in there, and I've had traps where I've gotten 50 to 70 moths per night. Once you get that many moths in there, that fumigant does, I mean, it works eventually, but a lot of those moths, they're bouncing around, they're hitting each other, yep. Scales are flying off. It's really hard to identify them. And I've caught most of the Spodoptera species that's in Florida. I've caught in my traps as non-targets. I mean, not commonly, but even some of the bigger Spodoptera species I've gotten. So, they're fairly specific, but not absolute.
Okay, excellent. And another question here, and I see you sort of answered it, but I'm going to just repeat it for everyone. It's we've tried various fall armyworm lure, but it did not attract fall armyworm in the field conditions of Pakistan. It attracted other Spodoptera species as well as other species. Why? Can you explain that to everyone?
Yeah, that's a good question, and it's funny. I've had researchers in South America tell me the same thing, that they've used a lure that I've used for years that's very successful in the U.S. and they don't catch a thing. And I don't know exactly what the reason is. I don't know if it's the age of the lure or if the lure has too many components and it's almost confusing to the moth. Or if the traps are not right. I mean, there's something not right. And again, if anyone wants some of the U.S. lures, I can certainly send them a couple just to test and compare. Chemteco with
Okay,
Sentry with Trace A or something.
you may
Also, the location is important. If you have your traps under trees or things like that, you're disrupting the airflow and the moths are not going to find the traps.
Oh, that's interesting. And you may regret sort of offering that because you might get a whole lot of
I know.
requests now, Rob, but it's very kind of you. But actually, we would be interested to compare some of those in Southeast Asia. And I'm sure Dr. Phoung as well has already been doing some work there and would love to carry on that sort of analysis and comparative studies. But yeah, what was really interesting, you mentioned how you'd looked at sort of the other places where fall armyworm can hang out, and you'd noticed that actually it's not just maize that they're in, and you'd collected or trapped them in other areas, and I think that's an important point for Southeast Asia. Where are these fall armyworm going when there's no maize around? So is that, I guess you use traps in the same way. You're saying it needs obviously in a clear area out there, but is that an important part, do you think, of fall armyworm management?
Well, especially in a region or in a district. I mean, I remember when the farming worms first started showing up in Africa, and one of the scientists there was saying, yeah, I go out of the maize into this grassy area, and there's just lots of fall armyworm, and I don't understand why. And I said, well, what's the grass? And it was a Cynodon. It's what we call Bermuda grass in the U.S. That's a huge host for fall armyworm. And so all this Cynodon grass was around, essentially encircling the maize field. Well, even though that was primarily rice strain, though rice strain love corn, they love maize. So anytime they harvest the maize and then get ready for the next season, the fall armyworm were just waiting to come in from these outside areas. So, and obviously you can't control the moths in those areas, but you have to at least know that they're there. And
Yep.
So by putting a trap in some of these areas to get kind of a seasonal, is there a seasonal pattern to them that might help the farmers know, oh, if it's Tuesday, they might be coming in or whatever.
Yeah.
Comparison.
Good point. Here's a question from Freddie. He's asking if light trapping is an option. Any views on that, Rob?
Yeah, before pheromones were developed the real commercialization of pheromones wasn't really until the 1980s. So all through the 60s and 70s, they used light traps. They were never tremendously successful for fall armyworm, as compared to say like Helicoverpa, Heliothus, some of the loopers, and it's not known. And a paper came out a couple of years ago, and I'll have to look it up to see, but they showed that the vision of fall armyworm, of Spodoptera in general, but Frugiperta in particular, is different, where they aren't really attracted to the wavelengths of most of the light traps. Now, yes, can you catch them in light traps? Sure, but certainly not to the numbers that you would catch, say, in a pheromone trap, which is interesting. And again, I can find that paper and send it, but it was really nice work. It's like, oh, Well, that's why. That explains a lot.
Yeah, no, that's interesting. And I'm saying interesting a lot because I am actually I'm finding this quite fascinating, and I can see other people are as well. We've got lots of questions. Now here's a question actually also from Freddie, and this has come up a few times in this region, and that the strain of fall armyworm. What about a hybrid strain? Would that make a difference to the pheromone or the lure that you use?
Yeah, that's the $1 million question. And so in the Western Hemisphere, we're pretty convinced there's a C or corn strain and a R or rice strain. So some of the work done in Africa and in Asia has shown almost entirely either C strain or hybrids of C and R strains. And so that's why, at least initially, the only crops that were generally attacked seemed to be maize and not grasses or rice or things like that. Now, I know more common now, rice is being attacked. So I'm suspecting that our strain is in Southeast Asia. The other thing that's interesting is there's a lot of really good debate on how many introductions of fall armyworm there's been in the Eastern Hemisphere. And so you have everything from, 1 or 2 to a bunch. And did it come from Africa and then go towards Asia? Did it actually get in Asia first and then get into Africa? So there's a lot of, I don't want to say controversy, but a lot of really good science that needs to be done here in the next couple of years to.
But does it does it make a difference to the pheromone, the combination of?
No, there's the chemical ecology group in Jena, Germany has done some really nice work. There seems to be differences that the females are putting out, slight differences, and it's not in components, but it's in the ratio of the components. But what we think is that the males can't tell the difference. That the evolution of the strains hasn't gotten to where the males, there's a lot of European corn borer is a great example, Strinia. There's one strain, I guess, that is 99 to 1 E to O, and the other one, E to Z, and the other one is completely opposite. So they can't talk to each other. Fall armyworm is not anywhere close to that. So we don't think the pheromone, that there's a big pheromone difference between strains. There might be technically, but I don't think the males can tell the difference.
Okay.
Which I could say a lot more, but I won't.
Me too. I've got a question here from Pierre. Bonjour, Pierre. It's nice to see you again. Rob,
hi, Pierre.
What do you think about the differences of the number of moths trapped per night between USA, Florida, and Vietnam? I'm not sure you would know because I'm not sure, but I guess you've seen a little bit of the presentation before. Any thoughts?
Yeah. What's difficult there is, unless you have the same exact trap and the same exact lure to make the comparison, then I don't think it's easy to tell the difference. It looks like, certainly in some of the areas that Pheung was trapping in, you have good numbers of moths there. But again, until you're comparing apples to apples, so to speak, it's hard to really justify that, oh, there's more moths in Florida than there is in Vietnam. So it's a good question, but until you do the comparison with the same trap and lure, I don't think your answers are going to be good. And plus,
so Dr. Pheung will send you through some of her lures, maybe, Rob. You can try it the other way, perhaps.
And plus, the seasons are different. In the U.S., fall armyworm is a migratory species. So the only areas are South Florida and South Texas. Everywhere else, even here, I'm in Gainesville, Florida, which is northern Florida. There's 2 or 3 months of the year I can't get fall armyworm. It's not here. And then it reemerges in April and May. Whereas in Vietnam, I'm sure they're around all year round. I can't imagine that there's not a population somewhere. So
Yep. Very good.
that affects trap capture too.
Okay, very good point. Here's a good question here. Are there any semiochemicals to attract natural enemies like parasitoids to enhance the parasitization on fall armyworm?
Yeah, there are. And there's a real debate on if they're effective or not. I can't, linalool is one of the chemicals, LINALOOL. And there's a couple other chemicals that are broadly known as green leafy volatiles. Those seem to attract parasitoids. What we don't, so we know like in a lab or in a flight tunnel, it works, but in the field, we think that they're not volatile enough, they're not lightweight enough to really get into the field and attract parasitoids in. So again, the jury is kind of out, and there's several labs in the US that are working on that, and actually I haven't seen work in the last couple of years. So my guess is, again, it worked in the lab, but wasn't overly effective in the field.
Okay, great. Just maybe one more question here around, does temperature reduce the efficiency of the pheromone?
Yes, definitely. I mean, it's a chemical, so the higher the temperature, the more volatile the chemicals are, the further they'll probably move. If it gets chilly, which for me here in Florida is a relative term, those chemicals are not going to come off the lure that much. So you're going to get a much lower. Plus, the moths don't like to fly when it's chilly.
Yeah.
I don't know, 60 degrees Fahrenheit, 50 degrees Fahrenheit. It's a combination of the volatiles not coming off of the lures, and the moths just,
Okay.
say, hey, well, sit in and watch TV tonight and go out tomorrow night or something.
Yeah, I can see that happening. I've got actually two more questions I'm going to ask you quickly. One around, you mentioned at the very start around the color is really important of the trap, and you mentioned green traps don't attract bees as much. Are you saying the other traps are attracting the bees in bigger numbers?
Certainly the standard trap, and I made a mistake once, and I didn't know I was near a bunch of beehives. I was kind of on the other side of that, and I didn't see them. So I had my standard traps out, and I caught half a bucket of bees, of honeybees. And I feel horrible about that, because those are insects you're not trying to get rid of. But I think bumblebees and honeybees, I think they're attracted. I think they think it's a big flower is the only explanation I can think of. There might be odors coming off of the vapor tape also that are attractive. But anyways, yeah, bees are a real problem in some areas. So the all green traps are really, if you're not looking for a species, or if you're not looking, if you're looking for general populations, the green traps are actually very useful. But most of the work that I do, I use the standard traps.
Okay, so one last question, Rob, and then I'm going to actually ask Dr. Pheromones just to come in and answer a few remaining questions. But you said where you put the trap in the field is really important. So maybe 3 or 4 tips of where you put the trap and where you don't put the trap maybe would be helpful.
Yeah, that's a good question. In fact, I saw something in the, I guess it was in the chat, that said that one of the problems is a lot of their traps get stolen. In the US, my traps don't get stolen, but they, kids go out drinking with shotguns and they shoot up the traps. So I've had that more of a problem than anything. I like to put them along field edges, so it's relatively easy to get to, but off of a main road. So that, that again, they don't get shot up. You have to be careful with the farmer and, not put them where they're going to get run over by field equipment. And again, now this is US agriculture. It's not agriculture in other parts of the world. So perhaps that's not a problem. But I've had I don't know how many traps ruined by a guy in a combine just chopping them up. You don't want them near trees. You don't want them near like a tree, like a hedge area or a tree row or anything like that. If you can get them in the field itself, that's great. But again, you have to be careful that the grower is not going to come in and put fertilizer down or irrigate or whatever. That's about all. I mean, you don't want them all together. You want to have a good 30 meters or more, maybe 40 meters in between traps. So you don't want them all bunched together. Generally, they say one trap per acres or per 2 acres. So what's that? About 1 hectare or something like that. If you're doing field monitoring like a farmer would do. But it's just, you have to combine ease of getting to the trap with, that it's not a nuisance. Because anything, if I'm a researcher and I make it hard for my cooperator to do his field duties, I'll never be allowed back in that area. So
yeah. Excellent. Okay. Hey, Rob, what a wonderful presentation. Really, really informative, and just fantastic to share all that knowledge. So we really, really appreciate it, and hoping that you keep safe out there from those, is it an alligator or a crocodile? I wasn't quite sure. Probably
mostly
alligator.
alligators.
Is it alligator?
Yeah, we do have crocodiles, but mostly the alligators.
Okay, well, careful those alligators out there protecting those traps. I hope they're on your team rather than working for themselves to protect those traps. But
well, I am a Florida Gator, so that does help.
Fantastic. No, absolutely great to have you share your thoughts with us, and really nice to see you. So I'm sure everyone appreciated your talk. Thank you. And I'm just going to introduce Dr. Phoung, if you could just maybe come back on, if you could unmute yourself. I don't know if you have any questions for Rob, but I'm sure you would send some of your lures as well to Rob to test. Could be interesting, and vice versa. So we might have to have a bit of an exchange in Southeast Asia and have Rob come down and see what we're doing down here. Be very good. Phoung, I've got a question for you. A couple of questions that people asked. Are you using your traps just for monitoring? Or are you also doing mating disruption? Because you did talk about a bit of that at the start, the 3 sort of different ways of using traps. What are you solely or are you solely focusing on one at the moment, or are you doing a combination of different uses of pheromones?
The first that's the pheromone currently we apply for the first is monitoring, and the second is the mass trapping. Mass trapping. Yeah.
So you're mostly on monitoring and mass trapping.
Yeah.
Excellent. Great, thank you. And I was sort of interested. Do you find in your mass trapping, I guess, and in your monitoring, are you getting many other non-target species? Are you getting bees, for example? We were just talking about that, or are there other? What are your most other common?
From my field test, the non-target species in the pheromone trap was very slow. 1 or 2 individuals per one trap for whole of the 30 days. Not too much non-target species in the pheromone trap.
Okay, great. Not too many non-target species. Okay. Yeah. And the other question was that we talked about with Rob was, I've got a question for you. Are you using them to monitor fall armyworm in other areas of Vietnam? So not in the maize, but in the non-maize areas, just to see where fall armyworm is hanging out.
The fall armyworm none on maize. Currently, the fall armyworm almost on maize in Vietnam. Yeah. On the party line, not too much, and we can control when the fall armyworm on party line.
But in natural areas, are you trapping in natural areas just to sit, like
We already try to place the pheromone trap on the pasteurized to collect the male model flora mealworm on the pasteurized field, but we cannot collect the male moth as much as in the May field. Not too much. Yeah.
Okay. Awesome. Hey, thank you, Pheung and Rob. Thank you so much for joining us today. It's been fantastic. I've really enjoyed it. It's actually really pleasure for the last of these sessions to have you both as guest speakers and real experts and really passionate about your field of research as well. I know that, and I can see it in the work that you're doing. And we've really enjoyed it. You've got lots of hearts and claps and everything coming up there on the screen. So, you know, really, you are very much appreciated. So thank you very much. We'll keep a recording of that and we'll share it with everyone. And I'm sure there may be some questions, and you may actually get some outreach there, both Pheung and Rob, around getting hold of some of those lures and testing them in other places. So thank you for that. I would just like to say a big thank you to everyone who's joined us across the whole series, all the other speakers across the total of 7 sessions. Fantastic speakers. The level of expertise is so high and so generous to share that with us, that experience. Also, thank you to everyone who's joined. There's some of you that have joined many of these sessions, and I've got a bit of a poll here if I can launch it. It's just an end poll, and I would really appreciate it if people could answer it, because I'm kind of interested to see how many people have followed these sessions and whether you enjoyed them. So really like to get your feedback on there. If you're still here, please answer the poll. Please keep safe, everyone. A happy end of the calendar year, and also a happy start of the, well, actually end of the Lunar New Year is going to be next, coming up end of January. So I won't see you before then. So happy New Year and happy Lunar New Year next year. It's been fantastic to have you along for this whole journey, and we really appreciate it. Thanks to Tamal today for helping out in the background. Keep safe, everyone. And if you've got any questions or you want to reach out, you can always email me. I'm happy to talk about anything with fall armyworm and IPM. It's an exciting project, and I'm really glad to be on board. So looking forward to next year. Thank you very much. Bye.